first minute of your presentation, I would like to introduce the paper being a particular organism that falls in this category. So let's say you take the Cyanobacteria, for example, that's the genus. That's your paper, right? So you'd say today's article has to do with Cyanobacterium, and this is a lithoautotrophic organism, and then show the energy system from your notes for the class. So everybody knows now exactly which one you're going to be talking about, right? Then make the transition to the paper. So if that doesn't make sense to you, then you're not where I need you to be. Basically what you're doing is you're taking a nutritional type, you're finding a research article, and you're beginning an introduction. So this is what you're going to talk about. This particular organism, this is the system right here. Okay, this is what we call it. And briefly describe it in 30 seconds. You know, and then because we're going to describe it all today in detail. What you're going to do is just introduce and remind the class this is the one we're talking about. Okay? Because there's 5 of them, right? So you're going to pick one of them. And the paper's going to be talking about one of them. And so the one you pick is the one you're going to briefly summarize. And then once you do that, boom, right into the paper. And that may talk completely about something different, which I hope it does. Such as, for example, you know, if you go into Cyanobacterium, you say, okay, this is a lithoautotroph. You know, this is how it operates. You can show a nice picture of that here. But then your paper might talk about some of the interesting applications of cyanobacteria today. And there's a whole variety of really interesting organisms. For biofuels, this is how they tap into this to make ethanol. It's the future. You might want to talk about that. Another example would be it's a way of producing electrical energy. So there might be a really interesting article about that as well. Okay. So that's kind of what I hope the articles you pick are really interesting applications or ways in which these organisms contribute to real life things that we can relate to, right? That's interesting to me. But it's really important that when you talk about something like that you begin your presentation by saying, okay, we talked about 5 nutritional types, and we're going to be talking today about this one. 30 seconds, this is what it is. Now, it's really interesting because the applications now can be used to produce ethanol. It's a way in which you can do this. So instead of using corn to produce ethanol, which is really difficult to do, right? Because you have to have lots of land, you have to produce lots of corn. It's really inefficient. Here you grow bacteria. A lot of times it's a eukaryotic called algae, but cyanobacteria is still a good choice. Produces ethanol. Okay? And that's the future, right? So that's what I'm hoping you guys can do, is pick one of those nutritional types. Another example would be the 5th one on that list, which is the Camel, Organo, Heterotrophs. Those are pathogens, right? So if you're really interested in a certain bacteria that causes a certain type of infectious disease, it's probably going to be a Camel, Organo, Heterotroph, right? So you could say, okay, I'm picking Clostridium perfringens. This produces a toxin, causes lots of foodborne diseases, causes gangrene, and it's a Camel, Organo, Heterotroph. This is how it operates, 30 seconds. And you jump into your paper, right? Okay, here's the organism. It's, you know, whatever that might be. Okay, so I haven't looked at your paper yet, but that's what it has to be, and that's what I would hope you do.
Quick question.
Yeah.
When you're searching for scholarly research on Google or wherever, a lot of times I was up to struggle to find results for, say, photoautotrophs. So I find results for photoautotrophs. So they do list specific bacteria and research that they use. So since it doesn't actually specifically say that litho, would you rather, you want us then to, I assume, research that bacteria to figure out whether it's legitimate or
Absolutely. And sometimes a good research article in the introduction section will tell you. And that's why we push this so hard. Is this is how we communicate as microbiologists. We say, okay, we're describing here a photolithotroph, and just go on with your presentation. Right? This is an example of a really unique chemo Organo heterotroph. And so that's how we communicate. So it's really, so a lot of the articles you'll see, it's a good article. It's talking about the metabolism of these organisms. It will define it as that. So you'll be able to catch that. Because the title may only say the bacteria, right? And as you said, it may not say chemolithotroph on the title. If you read the introduction, it'll probably say if it's a metabolism paper. Boom, there it is, and then that's what it is. So then you're good. Because if you really like the article, any article about any bacteria is going to fit in one of these 5 categories. Right? So just pick an article that you really like. Another way to approach it, say, I really like this bacteria. And chances are most people in this class are interested in infectious disease, so I'm not going to have both people pick chemo Organo heterotrophs, because that's probably what you'll end up picking if you like infectious disease. Right? So if one of you happens to pick one, then that's what it is. But then I would expect the other group to pick another one as well. Okay? So that's another way to do it. You're going to pick one, whether you went after it specifically or not. But the bottom line is whatever you picked, you can either research it, as you said, and find out what it is, or you can maybe find it by research and then get more specific and say, it's this nutritional type and this is the specific bacteria, the genus species that we'll be talking about today. That type of thing. So there's a lot of ways to do it, but the bottom line is that everybody will walk away from the presentation saying, oh, that was a really interesting talk about photolithotroph. I kind of enjoyed that, that type of thing. So that's what it comes down to. Okay. So good questions. And I'm going to press this a little bit more with all our presentations. And I'll remind you of this. Is that when you begin your presentations, all our topics that we talk about, when you pick those papers, are related to what we talk about in class, right? So I would expect you that when it's your turn as a group, you've got your paper up here. That paper probably will have a lot to do with what we just talked about in class. So that first 30 seconds of your introduction should be, today our article is pertaining to, precursors. And so what is that? To remind the class what we talked about in class, this is what it's about. And so this is the article we selected, right? So you're making that connection. So it's not something we do that's just sort of out there. It's directly linked to exactly what we're talking about in class. And it's really important you just remind the class, okay, remember that slide that was in our notes? That's what our paper's about right there. Okay? Boom, there you go. And everyone's like, sometimes the light bulb goes off and you're like, wow, I get it now. Okay? So that's what I would expect for the groups moving forward to be able to do that. Remind everybody of a key piece of slide is the best way to do it from one of these presentations. Pull one out. That's your very first one. And this is what we're talking about today. Okay. All right. So I'm going to add this to it. You guys don't have this in your notes. So basically, what we're going to do is really try to understand systems and be able to describe those 5 nutritional type systems. Okay. So that's the question, the exam question essentially. Okay. So we have 5 systems. We're familiar with some more than others. So the best way to begin to learn this would be really attach something that you understand and have heard about already to these systems. That's the best way to do it. Okay, because there's 5 of them. So in other words, let's start from the top and say, okay, a photolithotroph, number 1 on the list, right? If I throw that out there and you have no idea what to connect that to. There isn't anything solid in your mind saying, well, I have no idea what that is. It's something, some kind of bacteria, and it's a system it has, but then that's it, right? So let's do that. Now for me, when I learned this, and a lot of people are the same way, is that we all know and heard about this phylum of organisms called Cyanobacteria. Right? So if you heard about that, this is one way to learn this first nutritional type. Cyanobacteria are excellent examples of this type of nutritional organism. Okay, so what's a cyanobacteria? So the connection would be, number one, it's really interesting because number one, it's a bacteria. It's prokaryotic compared to eukaryotic cells, and it's photosynthetic. Okay, so of course, when you compare prokaryotes and eukaryotes, photosynthetic cells can either be a eukaryotic plant, right, or algae, complex cells, or they can be simple cells like bacteria that are prokaryotes, but they also have photosynthetic systems as well. And what's interesting about cyanobacteria, and this is why I think you would remember it, there's a lot of reasons, but number one would be it has photosynthetic systems that resemble plant photosynthetic systems. They're very closely linked, very closely. And because of that, they attach this bacteria in terms of evolution, in terms of it being responsible for the development of plants. Okay, so think about it. A plant cell or algae. If you take a single cell and you look inside that cell, you're going to have another organelle inside that called a chloroplast, right? And so if you look at that chloroplast resembles a cyanobacteria cell. Looks just like it. Okay? So what they hypothesized is that, billions of years ago, that this bacteria infected a eukaryotic cell, and that cell and that bacteria remained inside that bacteria or that cell, and then ultimately that cell developed into a plant cell, developed into an algae organism. There's an interesting organelle inside of it called the chloroplast. And so both of them contain inside of the chloroplast and inside of this cell called cyanobacteria have these systems that are associated with photosynthesis. Okay, and so that's the connection you should all make to thinking about this. Okay, if you're trying to remember how to do this. And as I said, there's some really interesting applications that I talked about earlier in class. That would be another connection too. Biofuels, etc. Okay, they all fall in this particular category. So what I want you to remember is now get a little bit more detailed about it, because when you talk about, notice there's two different photosynthetic nutritional types. Okay, the photolithoautotrophs and the photoorgano-heterotrophs. Okay, so now obviously they both have photosystems, but there's going to be a difference. The first one, as I said, resembles a plant, identical, right? And what we mean by that is it has two systems like this. We call these photosystems 2 and photosystem 1. And it's kind of funny, when it goes from left to right, they discovered 1 actually early on, and then they thought that was the only one, and they discovered 2 later, and they found out that the sequence actually starts with 2 and goes to 1, so it's all confusing. So somebody asked that question one time, why in the world is it 2 and 1? That's what happened, the way in which they were discovered, right? So anyway, so you got 2 and 1. And so, as I said, it's a photosynthetic system. It's a lithoauto, so that means it's organic. Photo means it's light, right? So light generates this energy. A couple of key pieces here that one has to understand. So when you look at this, what's interesting and when you need, and what's to remember about these photosynthetic systems? What comes to mind when you look at that? There should be some signature pieces that you just immediately look at and say, okay, that's important, that's interesting, I need to know that.
Think about the electron transportation as well.
Anything. That's one of them. I mean, that's important. There's another one that comes to mind that's probably more applicable to you, I would think.
Reducing power goes up as you get your photosystem more.
That's good. Very technical, but I doubt everyone thinks about that. But this is more practical, more. Just think about it. what's a real event happening here that impacts you? Oxygen, exactly. So take a look at that right there. This is like a plant. This produces oxygen, right? So cyanobacteria, it's another really interesting thing where it's linked to the ecological development of what we know Earth is today, right? When we talk about how the atmosphere used to have no oxygen, right, billions of years ago, and then at some point oxygen became part of the atmosphere, right? They think this organism played a role in that, right? So where does oxygen come from? Photosystem here. And the product of that is water, oxygen. So that I want you to remember, because it's just like a plant. And the second one is that these systems, 680 and 700, those P, I want you to know what those are too. Those are pigments. And they're pigments that absorb light at a certain wavelength. So at wavelength 680, this pigment, and these are what we call accessory pigments, they help gather wavelengths that are very close to 680, and they sort of corral them, if you will, so it maximizes absorption of light. And ultimately what happens is it triggers an activation to a new level of energy. The electrons move to a new orbital level, right? The chemistry is. We call that activation. And when that happens, it changes a molecule's redox potential. And we've talked about redox potential, right? Every molecule you can assign it a particular redox potential. That means, does it now have the ability to donate an electron, or does it have an ability to accept an electron, right? And you can change these pigments activations by having this light energy allow this to happen. So now you can see redox potential value on the x-axis. So it's jumped quite high. So it's very positive, right? And we know the flow of electron goes from positive to negative, right? Based on that chart that is in your notes. So now it's going to go and start to deliver this electron to the next molecule that has a little less of a positive value going toward the negative, right? And so that's the natural flow until it reaches here. Now, when that happens, it creates a really interesting, somebody said that the proton motive force or the electron transport chain. Now, because this is part of a membrane system, remember, that these systems are in the membrane, so basically what's happening is as electrons are being transferred, you also have this proton gradient that's produced as well. Very similar to the electron transport chain that we think about in pathogens, E. coli, for example, right? So now all these hydrogens are on one side, you produce what we call a proton motive force, and all those hydrogens then make their way back across the membrane through a protein called an ATP synthase, right? And that generates chemical energy, ATP. So that's how light now goes from light energy to chemical energy during this cycle process. And of course, what's interesting about that, it's got this second photosystem, the pigment again, it absorbs light at a different wavelength. Again, the same thing happens, redox potential changes through activation, and then those electrons are then transferred to some more electron-accepting molecules. Okay, and I do want to say, a note here, because the people that are really looking at this will catch this, that if you look at FAD, what they're saying is FAD delivers an electron to this NADPH becomes reducing power. If you look at that chart, FAD is actually more positive than NADPH. So you're saying, well, that goes against the rule, right? You're absolutely right. What makes it different, of course, your textbooks don't talk about this, is that FAD typically associates with another protein. And so when that FAD associates with another protein, the redox potential value changes. So it becomes more negative. So therefore, it now has the natural ability to transfer the electrons to the FAD or the NADH, producing that last step, right? So it makes sense. So that happens a lot of times. Now that chart they give you is just simply the raw molecule itself and the redox potential value. But if you start putting those, attaching them to a protein, that redox potential value can change. This is one example. These FAD things they're always typically associated with proteins. We call them cofactors, and that allows them to do what they're assigned to do. So anyway, that's interesting there. So anyway, it generates this, and why does it do that? Remember, the reducing power is that example of we talked about carbon source, energy source, electron source. This is the electron source. What's the definition of electron source? It's the ability to generate a reducing power. Why? Because that's part of biosynthesis. Okay? Biosynthesis. You have to remember that. Okay? Of course, a good way to remember that would be if you take a look at some of your diagrams here where they show, if I have it here, this one here. Electron source, making reducing power. It then supports this process right here. This is biosynthesis, or we call this anabolism, right? Metabolism is this, making ATP. As cells become parts of the cell and eventually makes the cell, you have to have reducing power that supports that. That's why they have these systems called reducing power systems. Okay? So when you go back to our example of this cyanobacteria, it has to have the ability to produce the reducing power, because ultimately that's what's going to allow it to then make that new cyanobacteria cell. Okay? So that's the connection. So that's how you would define and think about the very first example, the photolithic autotrophs. Okay, number one is part of a group of bacteria in the phylum called Cyanobacteria. Don't confuse that name thinking, well, Cyanobacteria is the name of the bacteria. It's not, it's a phylum. It means it's a broad umbrella that covers lots of different kinds of bacteria. Okay, sometimes people think that's what it is. Tca Cycle, that's the genus. It's an example of a very specific type of cyanobacteria. And there's lots of them out there. And they're actually one of the most diverse. So if you think about it and ever thought, well, you know, of all the cells that are out there in nature, cyanobacteria are right there on top. They're prevalent almost everywhere. And of course, a lot of them are associated with water. And of course, water covers most of the earth, right? So they're also found in salt water, so they're everywhere. You can actually take satellite images of the Earth, and you can actually see these green blooms. They're not always algae. Sometimes they're cyanobacteria blooms. They're actually green, and they are really important for generating oxygen for our system. So that's the connection. Make sure you understand. Number one, just remember a really good example. Give me an example of what they are. Cyanobacteria, that's what they are, and then give me some details about its processes. And this would be a good one to remember. Two systems, makes oxygen, always produces a reducing power, follows the rule of the values, because they don't always follow this rule. Sometimes they have to do something really interesting to get that. Okay? The second, again, same concept. If this comes to mind, first thing you should do is say, I need to think about an example of what type of bacteria that would fall within this category. A good place to start with is the soil and the water. Okay, so that's sort of a continuing theme, right? Soil and water are just full of microbes, and they're full of different kinds of microbes. So a lot of the soil and water that if you just go outside and have the ability to see what's in there, chances are you're going to find some sort of photo organo-heterotroph. The second, nutritional type, quite prevalent. But water seems to have a lot of these. Marine water, the ocean, fresh stream water, just full of this stuff. And what's unique about it, and different, but there's similarities. Number one, it's a photo, so it has to do with light. That's the same. What's different is now that its ability to obtain a carbon source is no longer inorganic. It's not CO2. It has to be an example of some sort of organic molecule. And so if you think about this, you need to be thinking about where does that organic molecule now enter the metabolic systems? Well, typically it'll enter glycolysis, right? It doesn't typically happen here. With bees and some other organisms, where would you think it would enter? If it doesn't enter glycolysis directly, where would it go? How would it put this organic molecule into the system? Because it's not relying on CO2. That's a CO2 fixing system. Okay, in plants that happens in the Calvin cycle. In bacteria, there's some other fixing systems that are unique. Now we're talking about organic molecules. Where would it enter? That's essentially what we're saying here, right? It's not relying on CO2 anymore, it's relying on organic. And that's what drives this whole thing. Light converts energy, and the source of carbon is organic. Take a guess, where would it be? I need you to know this, because this is going to be the difference right here. Follow the flow of metabolism. Go from glycolysis to what?
Tca cycle.
There you go. Now the Tca cycle has all of these intermediates, right? Those intermediates are really important for starting points to make proteins, right? The intermediates in glycolysis make peptidoglycan, they make lipids, they make all of these things, right? And if you follow it into the Tca cycle, all those intermediates, malates, succinate, oxoacetate, all of those become amino acids. Okay? So that's where typically these sources of carbon will then enter. And the best example would be acetate. And the association would be acetyl-CoA. Right? You should always know that. You go from pyruvate becomes acetyl-CoA, and that's how it enters the Tca cycle. Bingo, right there. That's the way in which this acetate molecule, it's organic, right? Still uses light, but that's how it then inserts this in order for the system to work. That's what makes it different than what we talked about as far as the first example. Okay? The difference between a photolithoautotroph and a photoorganoheterotroph. It's the carbon source and how it enters the system. Okay. And of course, they always, these are really good examples that they always think about. Because remember, energy is always generated in a lot of ways. Chemical energy, right? Substrate level phosphorylation, or oxidative, which is the electron transport chain and the proton mode force. Two ways in which that happens. This should be a continual reminder to you that this is how energy is made. Okay. Sometimes that's missed. It's a universal way in which they do this. Okay. And then the second thing, again, is the photosystem. But rather than having two systems, 1 and 2, right, it only has one of them. So that's another difference. And that's this example. But it still has to generate proton motive force, and it still produces chemical energy the same way, okay, even though it's not depicted in here. Okay, so that still happens. It's just a very simple one system. But what's interesting about that is because it's a one system, one of the unfortunate events of this is that there isn't really anything built in the system that allows it to produce a reducing power following the normal flow of electrons. And that's hence that little arrow saying, okay, what is this reverse electron flow? That's the unfortunate part of the system that exists. So it has to have a reducing power, right? Remember, reducing powers is what drives biosynthesis, metabolism. So if this guy can't do it by normal cycling, then what do you have to do? Well, it has to reverse the system. And that's unfortunate because if you reverse the system, that means you have to have available energy to make this reducing power. And so what happens, and I didn't put it in your notes, but it's in your textbook, and I want you to look this up. Put this in your notes. I need to look up a figure that shows how bacteria, through a reverse flow process, makes NAD, pH. In that example in your book, it's NADH. And it shows you very specifically how this happens. It takes the proton motive force energy, right? Normally that makes ATP. But rather than making ATP, it uses that proton force energy. Now it uses that to generate NADH or NADPH. So it's tapping into some existing energy, and it's taking it away actually from the ATP that it needs. So it's not very efficient. But again, that's the only way it can do this, right? And so it's kind of a unfortunate way in which this happens. But the reason they exist is because they now can incorporate organic molecules. So what's in water, those organisms that are CO2 reliant, all they need is CO2, right? They're good. Whereas now water, we know water contains lots of organic molecules. So then organisms that live in water, and they can still generate through light that process, then what would be interesting is say, well, if they're living in water and they're using light, why couldn't they use organic instead of CO2 organic systems? They can. Nature created these kinds of organisms. Okay? And so they're really useful as they're growing in water, that if you happen to have organics floating around in the water, it's still going to tap into the light, but it's going to convert that organic material into new cells rather than relying on CO2. So it kind of fits that niche of cells in nature. And water has a lot of that. Okay? So that's a good way to remember that type of system. And the reason it's different is because of what we talked about here. Okay? And of course, remember the contributors. What's really interesting too about this, you might, is that number one is found in water, but also it's a way to think about how you can decontaminate or get rid of pollution in water. Remember, a lot of pollution examples are organic molecules, right? So there you go. If this thing uses organics in water, but still relies on light to do this, it doesn't matter what this organic is. If it's toxic to us, to the bacteria, it doesn't really matter. It's an organic molecule. It's going to convert it to something else. And that usually is fairly safe. And it can sometimes completely mineralize it. We use the term mineralize. You can go from like a 10 carbon pollutant to CO2. Right? So, but the bottom line is those organisms are really good at cleaning up water. Pretty good organisms. And they have some really interesting things too, like we found they're really resistant to radiation. That's kind of a cool thing, I think. Not all organisms have that ability. Obviously we don't. So there's something special about them. Some really interesting articles talking about how that process occurs. And then also I thought was interesting too was that they produce hydrogen. And there's a lot of technology that wants to use hydrogen as a fuel. So if these guys can make plenty of hydrogen, instead of making ethanol, right? Instead of having cyanobacteria make ethanol, why not have these guys make hydrogen? And we have lots of hydrogen running vehicles, right? It's free. All you gotta do is just tap into anything that's out there. Pretty cool stuff, right? So, those are really good examples to remember. So, oh wow, those organo, heterotrophs are really cool organisms. I need to remember that. That's what they do. So be able to do that. The third one, this one you should remember. This one has to do with, because you think about it, you probably hear about it in the news, how farmers struggle all the time with providing enough nutrients for their crops. Okay? And that's why they fertilize, is they provide the nutrients that sometimes is lacking. Well, what happens is this type of bacteria that we call Autotrophs, they live in the soil, and unfortunately they take away, if you use all the different types of elements that plants need, nitrogen as an example. Plants need nitrogen. So whenever nitrogen is in the soil, the normal process of providing nitrogen to plants rely on bacteria. Okay? And it's a nice cycle that happens when it works really well. And that's this cycle right here. Okay? So you've got all kinds of examples of nitrogen, ammonia, nitrite, nitrate. All of these ultimately will be fed back into an organic system like plants, right? Plants use that nitrogen to build more plants. And then plants die and they break down and release this again. So it's a nice little cycle. So what can happen is that these Autotrophs that are associated with agriculture is that this system, instead of going and producing that last nitrate molecule, there's different bacteria, instead of giving it to the plant, it actually convert it to nitrogen gas. So now the plant is lacking the ability, right? And so what can happen is, if you have too much of this particular organism that converts nitrate back into gas, then that plant no longer has the available nitrogen. And what usually happens too is that this is what's usually part of fertilizers, nitrate. So farmers are dumping tons and tons of nitrate in the soil, and what's happening is this bacteria is converting all the gas. And so your plants are not benefiting from that fertilizer, right? So it's a huge problem. And that problem has to do with the fact that these nutritional types called chemolithoautotrophs, that's a good example. They're part of that problem. Okay. And so that's one way to remember. A second would be, you guys are familiar with different types of mining, the mining industry, right? Coal mines come to mind. Coal mines have a pyrite example. There's a nice little article in chapter 11 I want you to read. So make sure you put in your notes. You need to read the acid mine article. That's an example of how a particular type of bacteria called a chemolithoautotroph. Okay. Basically what it does is it takes iron, which is part of pyrite, converts that to the ferrous form, and the ferrous form goes to the ferric form. And that ferric form is what fuels the bacteria. Remember, it's a litho, right? It needs an inorganic as a food source. Iron's the food source. And the product of that is sulfuric acid. And what happens is you've got these mine tailings that have pyrite, and this bacteria in the presence of this produces, it's fed because it has plenty of iron there, but it also produces a lot of acid. And what happens is you end up contaminating the soil and the water. And you get high levels of iron that precipitates out, which is very toxic, and of course the acidity as well. It's not a good thing. Okay? And so, and it's aerobic. And the way you can stop this is if you happen to have a way in which you can tap this area, take away oxygen, you can actually prevent this from happening. But again, that's an expensive thing to do. You can't do that, right? We're talking about nature. So there's a couple of ways to remember this. What's a chemolithoautotroph? Farmers will probably remember that. Or miners, or if you happen to live in an area where you got old mining residue still there, it's likely to have this type of contamination go on. And in Colorado, for example, it's a huge problem. They're trying to clean these up because a lot of these mines are abandoned, but what's still there is the pyrite residue. And it's just a normal reaction that goes on and creates a lot of toxicity. Not a good thing. Okay, so that's what these organisms, I think, would be known for when we talk about that. Just a couple examples. Soil, toxic rivers, that type of thing. Okay. The fourth one is what we call the heterotrophs. Again, same thing. Try to remember a good example of what this would be. I think the best one would be, we have put it in an example, that it's water associated. What type of water? Well, a lot of times it's marine water. It's not surface water, it's down on the bottom. And there's these examples of what they call hydrothermal vents. Okay, the bottom of the seabed, what happens is some of the Earth's core lava is spewing out some of the processes, right? And a lot of that release has lots of sulfur. So if you're a diver and you happen to go down and look at one of these sulfur vents, basically it's spewing out lots and lots of sulfur, just tons of sulfur. So then you say, okay, well, I know anything that's a some sort of litho organism is probably going to rely on an inorganic molecule. Sulfur is inorganic, right? So then you're thinking, well, if there's a lot of sulfur down in this part of the Earth, I wonder if there's any microorganisms that can survive on this. And the answer is yes. Because a lot of sulfur, okay, it's a chemolitho system, okay, and so it allows it to grow really well and utilize that system. And what's different about it is that rather than having the source of carbon being CO2, like the previous example, it's going to rely on an organic system. Okay, so that's your acid. And the same thing happens here, is that organic molecule is inserted directly into the Tca cycle rather than glycolysis. And that's how it supports this process. So that's an interesting thing about this particular type of organism. Okay, and that's what makes it different. Okay. Now I did do a little bit of rearranging and copy and paste some additional slides to support what I've just been showing you here. I'll send you this again, so to put it in a little bit of order from the one you got the other day, so it'll help you a little bit more. So it'll make a little more sense. And then, of course, the last one would be, here's this Tca cycle, right? So you got this, just to give you an example, of acetate comes in here, and it has this really interesting bypass system. We call it the glyoxylate system, which makes it very efficient for using that as a source of carbon. So you can read about that in the textbook. Okay. And what's interesting about that is, I think that a lot of times these organisms, what they discovered is that they tend to surround and grow in areas of high concentrated inorganic pollutants. Okay? So this is an example where you have a lot of inorganic pollutants in water, and they form these really interesting mats. So what they are starting to develop is that whenever you see these mats, these are good indicators of where there might be pollutants in seawater. So it's really kind of cool. It's just, why is that? Because they're seeking out an inorganic energy source. And so that's where it's heavily concentrated, and that's where they're going to grow. It's really kind of interesting and cool. And then, of course, the last one, you guys all should know this, so I won't say too much about it, right? When you first took this course, you first took biology, this is what you learn in metabolism, right? Glycolysis, TCA, electron transport system, aerobic, anaerobic respiration, and fermentation, right? That was the solid background. Okay? Lots of pathogens, E. coli, Salmonella, Clostridium, all those examples use these systems, right? So we won't say much about it, but I do want you to answer a couple questions about this that we started to answer on Monday, but it's going to be important for you to follow through. That we can think about comparing and contrasting these two respiratory systems. Okay, you need to know the difference. Of course, the big difference would be number one, the amount of energy. Okay, which one makes more energy per molecule? Aerobic. Okay, you should be able to tell me why. Just quickly, why would that be? Okay. Give me a more specific reason. Aerobic, anaerobic.
The chemical product of it is higher, or aerobic.
The chemical product. Okay.
The amount of kilojoules produced is higher.
Okay. Almost. Give me another reason. Give me a more clear reason why an aerobic would be more energy producing per molecule than an anaerobic system. You'll know it when I tell you, but just think about it. What's the answer?
Aerobes can use the electron transport chain, and fermentation goes.
So it's not fermentation, it's anaerobic respiration. So that would be a third category, which we'll talk about on Monday. But we're just thinking about respiration, two kinds of respiration that happen. But you're almost there. It doesn't use oxygen. And so follow that thought through, then you're on the dot. A little heavy. So when we say anaerobic respiration, means there's still an electron transport chain. Still there. Aerobic, the final electron acceptor is what? Oxygen, right? And that turns into water. And that's why we call it aerobic, right? Because that's the point at which oxygen is used. So if we call it anaerobic and oxygen is not used and it still has an electron transport system, then how would you define that now? Final electron acceptor is now what? It's not oxygen, right? It's something else. And that's the difference. And so whatever that is, if you now take the redox potential difference between glucose and oxygen, and glucose and, let's say, a final electron molecule is nitrate. This is glucose oxygen, this is glucose nitrate. Pretty small, right? There you go. That's why anaerobic respiring organisms produce less energy, right? So that's what we want to be talking about. So And if you follow this system all the way to the end, it has to do with the available proton motive force or hydrogen ion gradient that's produced on the side of the cell. And these are two examples of where you can see that process. So here's your nitrate entering, and then the end of it is conversion of gas, and here you've got oxygen being the final electron acceptor, and the amount of hydrogen that's on this side and here is going to be different. More here, less there, and ultimately when that rushes back into the system producing ATP, there's not as much there as well. So that's another way to describe why aerobes produce more energy than anaerobes, because the amount of proton motive force generated is greater. Okay? And you can sort of see that when you look at these systems. So we need to answer that question. Other one is this one. Just revisit it again. Why do this particular organism impact the agriculture industry? This is the way in which these organisms steal the nitrates and make the nitrates into nitrogen gas. There's all that fertilizer, triggers this whole process, ultimately releasing nitrogen gas. Okay. And then nitrates on top there as well. Okay. So the plants aren't getting the nitrogen they need because of this event happening right here. So that's another question that I would like you to answer. And the last one would be versatility. Okay. So when we think about these systems, whether they use organic, inorganic, there's also differences between examples of organic, right? So here's two examples. So the same organism, this organism can use glucose, produces a lot of energy, but then it can also use methanol. And so the question was, why does methanol produce less energy than glucose? Electrons, proton motive force, all of those things. But this is sort of how you could visualize that process happening. Right? Glucose will have that electron transport chain that has the ability to produce lots of hydrogen ion gradients at that point, whereas methanol enters at this point, therefore just produces very little hydrogen. So immediately right there you can see how the amount of gradient is different between the two, hence less ATP. Right? So that's the versatility. These organisms, this is the same organism that has the ability to switch from one to the other depending on the environment. It's kind of cool. And it's the same with a lot of these other nutritional types, that there are some examples of where if oxygen is present or not present, and that's the difference between the lithoautotrophs and the organo-heterotrophs, the first two examples. You can have one organism that can have both of those. So when there's a lot of oxygen, it does its thing, and when there's no oxygen, it switches to the other. So they're really, really interesting organisms, how they can kind of cross-utilize different nutritional type examples for the same organism. And that's the point I want to make, is that they're not always in that one category only. Sometimes you got these really complex organisms that we discovered that can share these different systems. And that's pretty cool too, I think, because it just has some wonderful possible applications that we could maybe use in the future. All right, so reminder of the groups. Pick an example of 1 to 5, and read it to me. We should have some good presentations on Friday. Okay? See you later.